Let's pick up where we left off.
Aren't you going to show the photo first?
Oh, fine.
There you go.
That is the first day of kindergarten at my school, everybody. For you guys who were not here for MCAT, I want you to appreciate these were the moms back in September 56 having a cigarette while they're taking their little kids to the first day of kindergarten. That's how times have changed. God bless you, Mrs. Combs. That's what the girls look like for Easter. None of these guys could figure out, okay, where I am in this. Where in the world is Lou Stein?
Very left.
Which point? Come up and point. It's your call. Hey, you're. You can't talk the talk unless you walk the walk. Yes.
You got it. She's good. You sat here the entire time trying to figure it out. I can tell it was you.
Bless your heart. It's all right. I'm only like 50 years too old for you. Okay, 45. So much for that. Now, I thought we get more mileage out of that, but I digress. Okay, here we go. So this is kind of where we were last time, and I know you've had a lot of this stuff before, but it's important because to me it's still part of my course that I have to do with regard to this. This is again is really right now we're dealing with the cellular response, and we're dealing with the second level of the innate immune system that's there. And again, we talked about it. Neutrophils first, macrophages follow, and then depending on whether or not it's pathogenic or an accident, complement, even some aspects of the adaptive immunity. And again, the idea, those factors generically referred to as cytokines, meaning making cells move, basically motivate both from the bone marrow and what are called body pools. So there's other areas, not exclusive, where they bring these fat, and that's what I talked about before with the blood tests. When they have to do this over an extended period of time, they begin to immature forms that are not nearly as functional or non-functional. And that's one of the ways we monitor just how effective the response is, whether it's appropriate, inappropriate, or excessive. So there's a lot of layers to all this stuff, as you might guess. The so-called leukocyte-inducing factors, they marginate because the chemicals basically interact and cause these cell adhesion molecules to effectively latch onto the elements, particularly the neutrophils, to start, and not the only. They diapodese because they can go through these now enlarged intravascular clefts, particularly at the capillary level. Okay, it's called positive chemotaxis. The monocytes arrive considerably later. Okay, they transform into macrophages. So they take over for the neutrophils that are spent. The neutrophils are not able to survive after they encounter a pathogen or damage because that's really their job. And so that's why when we see pus in an infection, a lot of people mistakenly think it's filled with bacteria. The reality, if it's filled with bacteria or microorganisms, they're basically dead. Okay, mostly it's tissue fluid and spent white blood cells that get, and to some extent, that's an important, clinical sense. People erroneously culture pus. They take a sample of the pus. It's a mistake. If you take a sterile gauze pad, remove the pus, you culture the sides and base of the wound itself, if you can, if it's external damage. That's where you're going to see most accurately what the organisms are involved that are part of it. That's how it's done. So this is the illustration we let this. This is basically getting some type of sharp object. Inevitably, it's not sterile, which means there's microorganisms on it or carried down from the skin surface. And because of that disruption of tissue, now you get the chemotaxis, you get the margination, you get the diapodesis, and eventually the interaction that occurs there. And they show you that. Leukocytosis, margination, diapedesis, chemotaxis. So you can see all of these things, and they sort of give you it's a nice little, to me, a summary slide that works effectively. Tissue injury, inducing factors, leukocytosis, migration, margination, diabetes, etc., until eventually pus may form from an area cleared of debris. And now all the inflammatory chemicals, they don't really, elaborate other than generically histamine, complement, various kinins. The one you hear the most about is called bradykinin, prostaglandins. And what does it do? Increasing circulation with arteriolar dilation and capillary permeability, attracting the white blood cells to come in, the fluid leakage, the localized increased blood flow is the heat and redness. As you can see here, the pain particularly from those chemicals, eventually, and nerve endings possibly impairing function, and all the leakage of fluids, particularly protein-rich ones, because they'll draw other fluid as they attract fluid, and almost like a colloidal kind of a pressure, and that's where the swelling. Remember, redness, heat, pain, and swelling, or swelling and pain, if you want to go in order. So here's pus or purulent drainage. Yellow might not, a lot of times the appearance of the pus gives you some ideas clinically, whether if it's blood tinge, whether or not there's blood vessels. We used ultraviolet examining light for some kinds of pus. So you'd be able to say if it has a particular glow. The reason why we see the creamy yellow is the most common offender by far, what I like to call public enemy number one, Staphylococcus aureus. And the aureus, the AU, see it up there? AU. Gold. Right? Not AG. Right? That's how it got names. Aureus. Gold and yellow. Whereas the staph epidermidis, which was originally called staph albicans, because alba was white.
Oh, like an albumin and an A.
Yeah.
It's all coming together.
And so AU, if you back in the early days in like 1964, one of the early Bond movies was from the third Bond book, Goldfinger. Bert Frobe, Sean Connery.
It's my second favorite movie in the series.
Honor Blackman.
Second best movie in my opinion.
I'm trying to remember the guy who was Odd Job. Great character. The hat. Great book. Love those.
What's your favorite Bond?
James. It could be Goldfinger. I might probably would root for that. I like Dr. No.
I'm a big Dalton fan, so I like License to Kill. That's my favorite.
Yeah.
Whoa, Dr. Stein, you can take a hike. Timothy Dalton's underrated.
Have you watched in 1923? He's a bad guy. In 1923, the Yellowstone the prequel. It just finished its last 8 episodes of this first season. Mm-hmm. Oh, he's a bad guy. He's a good guy. I don't mind. He was a bad guy in Rocket Man. Just saying. Okay, so abscesses. Collagen fibers are laid down and it gets walled off. The problem with abscesses, I say this all the time in micro, pus is lazy. It takes the path of least resistance. So if it's not walled off, it goes along tissue planes, and that causes what we clinically call tissue space infections. Those are bad. So you have to, that's why one of the most important things we do is it's called, if it needs it, it's called incision and drainage, or IND. You have to give the pus a pathway out. So that's really important. So again, some bacteria, tuberculosis is clear, but that's in the lung. Resist digestion, they remain viable. They form the granulomas, which is the surrounding of white blood cells. So it's surrounded by uninfected macrophages, forms a capsule. That granuloma, because it's firm, became tubercles. That's again where the name tuberculosis. So they can remain inactive forever. A lot of people have what's called latent TB. They don't have to be treated unless there's some underlying reason to do so. And you can activate, particularly when your immune system is compromised, or it could activate if you have a severe lung problem associated with, or a bad case of pneumonia. Potentially could do that. So it's a little more common than we think, to tell you the truth. The other things that are part of it that enhance the defenses and are technically part of the cellular, even the chemical portion, antimicrobial proteins. Okay, they can attack them directly or impair their ability to reproduce, so inhibit it. The most important antimicrobial proteins, interferons, which we'll look at, and we'll look at more than once, and complement proteins. Interferons, they're a group. Interferons are something that initially in the early days of treating things like hepatitis C, they genetically manufactured it. They were very, very harsh treatments. There are much more advanced treatments today. You see advertised on TV all the time for hepatitis C. Okay, they're there. So cells infected with viruses secrete certain kinds of interferons that basically signal other cells, okay, to stimulate production of proteins that block and degrade viral RNA. So alpha and beta work this way, and they also activate the cells in your innate system that are designed to attack these kinds of viruses that invade cells. So if the interferons are ineffective, that's where the NK cells come in. And again, we're going to go into that sort of laundry list of the distinction between things that we deal that are humoral and things that are cellular. It's true. To me, it's the crux of understanding how your immune system works. The gamma immune interferon made by lymphocytes, widespread effects, activates macrophages, activate NK cells, cancer fighters. So these are some of the things, and this is the one was genetically manufactured. Why do we not use it today? I probably until we got more sophisticated, maybe in between 2005 and maybe 2012, right when I started to be here full time. I usually had a student or two who was on, and they couldn't even function. Okay. One girl had hepatitis C. I remember had to have that. Somebody else I think was having that as augmenting treatment for MS. There are better means to treat it, but the impact was so severe as to be almost rendering that person really unable to function. Okay. Nowadays there are certainly better methods, more preventative in nature for genital warts, and we'll certainly go into that part of the unit when we begin to deal with reproductive physiology with regard to HPV and treatment approaches to that are there. Multiple sclerosis, why it responded to this is we think, and it's something we discussed in this chapter, in a lot of individuals, we think there's a viral component. There's a very high statistical correlation between levels of, I think it's antibody levels to human herpes virus 6, which causes a relatively innocent childhood disease that most of you have had called roseola. Okay. And about 90% of us test positive for it. But the levels that are very high, there seems to be a correlation coefficient. That doesn't mean cause and effect. Something's being studied. As to MS, MS is more ubiquitous than you think. There's a lot more MS going around than you think, and we'll get into that when I do the immune disease, which is part of this chapter as well. What we're going to do as an aside when we come back Monday, because you'll be all tied up with this test, basically lecture's going to move to lab, because I got to catch you up on cardiac and blood vessel physiology. So you're going to have to hear me blab for a while, which is a good thing. Voila. So here they're showing you the virus infects it. Okay, it replicates. And they show you what happens. The interferon genes are switched on. The chemicals extruded, and then basically it generates something. There's a receptor for it over here, and that causes interferon binding, stimulates cell to turn on its genes, for antiviral proteins. It's like, there's a virus in the neighborhood. Yeah. Complement with an E. 20 blood proteins that are always there. They're part of our blood proteins. And again, most proteins circulate in the inactive form. They have a variety of names that are there. I'm not going to go into it. Major mechanisms, because they play a pivotal role in things like opsonization, in things like disposal and endocytosis. They are facilitators. They are very, very helpful in that. So we need them. Okay? It actively enhances inflammation, directly can destroy bacteria. It interacts. And again, this is one of those wonderful mechanisms where when something triggers your innate defenses, it's basically sending a companion signal to your adaptive defenses going, wait a minute, there's a problem. So there's different pathways. You'll be happy to know I don't do these. Does Dr. Maurer do these pathways for the complement pathways? Good, because I don't want to do it. There's a lot of stuff. If you move on to graduate studies or medical school, if you go on to PA school or to some type of other doctoral programs, you probably have to memorize this stuff. I just think it's energy expenditure, and I never feel that people need to have an excessive amount of that. Hold on. Oh God. Oh, it was potential spam. I love that. Okay. Antibodies are the classical pathways. Antibodies first bind to invading organisms, bind to complement, double binding, and then effectively there's this cascade is triggered. There's something called the lectin pathway. These are produced by your innate system. Okay, when lectin is bound to specific sugars on invaders, it can activate complement, and another pathway called the alternative bind directly to the complement cascade is activated spontaneously, binds directly to foreign invaders, okay, and allows it to proceed. So there's these different pathways. They are important. I don't see the need to remember which is which. There's a sufficient amount of material in this chapter, I think. Each pathway has a particular sequence that's associated with it. And as you can see, initiates a common terminal pathway that enhances inflammation, so this is what they do. Promotes phagocytosis, causes certain cells to lyse or break apart. So they're to your benefit, okay? The insertion of these membrane attack complexes, okay? Effectively, it's this sort of interesting way, and we see this a lot, okay? And we go over the same thing in micro, the way that your body defends itself in the same kind of a unit, is this remarkable ability of certain things to basically, in order to get into the cell to damage it, you have to basically insert poke little holes in it permanently. And that's what these membrane attack complexes are like. Opsonization, okay? Mast cells and basophils, so it's like a co-amplifier, more neutrophils and white blood cells. So you can look at all these pathways and look at what they do, but effectively, it's not unlike clotting in that regard, that these pathways will all eventually lead to the C3. And once that does its job, then these membrane attack complexes form that insert membranes in what you're trying to attack, be it the cell or the entity. And it allows different things to, as you can see, creating pores that lyse the target cell by allowing water to enter and cell contents to leave. I love that. Again, when we look, fever is something that you want to be there when it's supposed to be there and not when it's not supposed to be there. Okay. There are certain, when fevers don't break when they're supposed to, or they go to higher levels than they should. You and I, as adults, have a limited capability to tolerate high. Children do not, babies do not. babies, 104 temperature, I say it's in every class, is not a ticket for a baby to be admitted to intensive care or to the hospital. It's not. Maybe I shared this with you. I'll share it with you again. The protocol for years at Children's Hospital was this. Maybe they've modified it since I did things clinically. When you came in with what's called an FUO, which is fever of unknown origin, okay, at Children's, the first test they do, let's say you have 104 fever, the first test you do is a urinalysis. So a baby comes in, they get a urine sample, relatively easy to collect. If there nothing shows in the urinalysis, they still are not doing a blood test. Okay? The next test they do is a chest x-ray because it could be pneumonia. Baby crying and screaming, it's pretty hard to hear chest sounds very well, but you might be able to discern them in the x-ray. So assuming those first two are negative, what do we do next? Oh, about time we got some blood tests. No, maybe you'll do them, but the next test that you have to do is a spinal centesis. You have to do a lumbar puncture. Because by this time, if you don't have a cause with a high fever like that, it's time to rule out bacterial meningitis. And the only diagnostic test for that is a lumbar puncture. And this happened to us, to my first wife and I, I always do this when we study rotaviruses, which is the infantile diarrhea virus. We foolishly, stupidly, the only word to use for it, took our recently adopted 10-month-old son to visit the couple that we were in the adoption groups with, we were very, very close with, down to San Antonio to party for New Year's. They do it really well by the Alamo. Just saying.
San Antonio.
In San Antonio. Riverwalk, yummy. Okay. And we fine for the first 2 days. We got there 2 days before New Year. Went down to the hotel, had a great party, Texas two-step, lots of good stuff, and came back up, kids throwing up, diarrhea everywhere. Everywhere. 10 months old. Rushed them to San Antonio, Santa Rosa Children's Medical Center. Looked at them first day, gave them some treatment, came back the next day, not a lot better. Guess what? Lumbar puncture. We're going nuts, 2,000 miles away from our pediatrician. Okay, turned out he had rotavirus, and they all had to do was treat him supportively. He was in the hospital for a week. We thought he was going to die. Right? It gives you an idea, and that's what is explained to us. Once that test was negative, it didn't take long for them to do that. Okay, long before they did blood tests. So it gives you an idea. So high body temperature. Now you and I get 104 temperature, they're going to want to admit you. Yeah, because we'd be prone to febrile convulsions, and it could cause damage. It could cause conceivably brain damage. That is systemic response to invaders. Leukocytes and macrophages exposed to these foreign agents secrete pyrogens, and there's a lot of bacteria that do that. They act on your thermostat and the hypothalamus. The good news: it increases metabolic activity, increases repair, increases the generation of antibody, all the things that you need to fight it. The liver and spleen sequester iron and zinc, which are trace elements that microbes need to reproduce. Okay? So that's the good news with it. The bad news is when it's unchecked. So that's really the nugget here. And then we start getting into and that really concludes the innate part of our immune system. And now we enter the realm of the adaptive immune system, which is, so those mechanisms, to summarize, were all non-specific. If they're a virus, they may respond more with NK cells. As a bacteria, with the response being more likely to be the traditional neutrophils. Macrophages are going to play a role in both of them. That's kind of the way we have a lot of sort of generic, to use the term, antibodies and antibacterial substances and secretions that are present all the time. So there's a lot of innate immune things. But what happens when it's not so easy and we've somehow breached the next barrier? Okay, now we get into the more powerful system that is really sort of, again, the linchpin when we talk about, and it's been endless discussions about immunizations and vaccines recently vis-a-vis COVID. This is the system that we're trying, by using those immunizations, trying to stimulate in a preventative sense, because we want to be sort of pre-encoding our internal protective hard drive, our defensive hard drive, with information to make antibodies so that we can rapidly respond to challenges. That's the whole ball game. So we're going to explore the science that's behind that. That's really the next part of this discussion. Are you happy? It's very specific. While it's specific, one of the problems with it is these immunological cells and immunological chemicals go out in tremendously large numbers all over the body. So I'm sure many of you, when you got, let's say, the second COVID or a booster COVID shot, you probably felt pretty rotten, I'm guessing. There's a lot of responses. The trouble with immunizations is they are not innocent. Okay? There's a lot of conditions. This is the anecdote that came from 1976. It was very famous. When I get to viruses and immunizations, I talk about that in micro to my students. We had a thing, they thought it was an occurrence of what's called the swine flu. So Spanish flu, which was a bird flu, the greatest killer of all time microbiologically, far more than COVID, far more, based on percentage of population, came from basically a novel virus that was created from a confluence of factors, one of which were viral particles from birds interacting with humans and other species. Pigs can do the same thing. So when I have a pig, we had one of these, an H1N1 in 2009. That was a swine flu variant that we thought could have been it. And it was a number of people, my middle one included, who got sick from it. In 1976, they saw a little cluster break out in some military recruits, and everybody went nuts. Whoever was in charge of the labs and the health services back then said, "Oh my God, we're having an outbreak. So we have to make a vaccine." And they were going to do a nationwide immunization program, just like they did for polio about more than 10 years prior to that. Wherever they started, for whatever reason, they started doing these immunizations first. I don't know how it got there first, in either Minnesota or Michigan or Wisconsin, up there. They got a cluster outbreak of what's called Guillain-Barré syndrome. It's a very well-known syndrome that accompanies not only viral infections, rarely, but also immunizations. It's an ascending paralysis. And after initiating this, they immediately had to stop it, that program. So the reason I'm telling you this for context, they're not innocent. So it turned out that when they crunched the numbers, that it was just a cluster and it was not unusual and not considered to be directly, and they might have just said that to avoid getting sued. Okay, but very famous. You don't have to know it all right now. It's one of those neat little diseases you hear about, and we heard about it again with COVID. Guillain-Barré syndrome. Syndromes mean we don't know exactly the cause, but we know how it comes in. And how people begin to ascend, paralysis, ascend, eventually respiratorily have to go on a respirator. Most of the time they recover completely. Sometimes they don't. I remember sitting in an in-service, basically a grand rounds at Cherry Hill Hospital, where we were doing a presentation on this, because it just was November of 1976, right after the bicentennial. So, moving on. Amplifies the inflammatory response, activates complement yet again. The big problem is our initial exposure takes time for that information to be encoded, and sometimes that's a problem depending on the severity of the infection. That's why for things that we know are deadly killers, diphtheria, tetanus, pertussis, why the determination was made to make sure that we continually be immunized for it. It is specific, very specific antigens. If the antigens vary, it's no longer specific. So we've started to see that. That's classically why we have to change the influenza immunization on an annual basis. Why we're going to have it's a cottage industry making COVID. Oh, we have another booster that's good for this. What's interesting about that, the severity of COVID and the potential lethality of it has gone down, but the immunizations have gone up. We've gotten to the point where it's starting to get into balance. Okay. It's systemic. It's not restricted to any one site, goes throughout your body. But what the key issue is, the memory. Mounts an even stronger attack to second and any subsequent exposures. That's the beauty, if it works properly. So a lot of ifs that are involved. The first thing you learn about dealing with the human body from a standpoint of a caregiver, be you a professional or even a non-professional caregiver, is nothing is exact. There's always exceptions to the rule. There's very few always. So the two main branches that we're going to look at first, the humoral, meaning it flows throughout your bloodstream and your plasma. That is the antibody-mediated portion of it. The second and the more complicated is the cellular-based immunity. And that's where we get into this distinction that I've been harping on incessantly. But it's my way. Antibodies, they are the way that we deal with humoral immunity. They're made by lymphocytes, specifically a large number of B cells rapidly change into something called a plasma cell and produce immense amount of antibodies once the systemic response ramps up. After the priming of it, once it starts to produce, oh boy, it produces. They temporarily target the cells, they inactivate, and what they do is in some way, shape, or form, they take these circulating particles, bacteria or viruses, toxins, things that are circulating, if you will. And that's the concept of humoral. It's not something, it can be on a cell, but not in a cell, or it can be circulating in the bloodstream or tissue fluids. And what it does is it inactivates it and identifies it. It tags it, for lack of a better word. And that allows your body scavenger cells to come on, read that as macrophages, to go, lunch. Again, channeling my Dr. Lecter, I'm having an old invader for dinner. So the targets by definition are extracellular. As opposed to cellular immunity, we have to have a cell. They directly kill cells that are infected. Or indirectly, they release chemicals, okay, that increase inflammatory responses, more of those penetrating, molecules that go there that we saw with regard to complement, activating other types of lymphocytes or macrophages. They have cellular targets. So this is kind of the way, if you wanted to draw it or think about it schematically, if it is a cell that has been invaded by a virus, a bacteria, okay, or a cell that has been altered by it, a cancerous cell, a transplanted cell, even a transfused cell. They're less so here in the transfused cells. But the classic example are cells that have been invaded by a microorganism, most commonly viruses and bacteria, cells that have been altered in some way, shape, or form, and those are typically cancerous cells or transplanted cells. Those all lead to cellular immunity. Everything else, humoral immunity. That's the way they work. They respond for years. The term antigen, and I told you a little bit about this with the blood unit, is the term was coined as antibody generating. Originally, we thought everything was antibodies. So anything that would provoke an antibody, we called an antigen. We didn't, our concept of self versus non-self was very limited. Now, any of those markers on cells that are effectively short chains of carbohydrates on proteins, for the most part, are considered antigens. Okay, so you have self versus non-self antigens. Okay, so substances can mobilize adaptive defenses and provoke an immune response, or would be, in this case, typically non-self antigens. Most are large, complex molecules not normally found in the body. Can be complete, typically. Sometimes, and these are interesting, are haptens, which is it's like code for half antigens. They are substances that are not antigenic until they bind to a protein, and then they become a complete antigen, and that's typically allergens. Those are typically ones that provoke allergic responses. And then, of course, we have the self-antigens that are there. So the complete antigen, immunogenicity, ability to stimulate specific lines of lymphocytes, be they B cells or T cells. Reactivity, the ability to react with those activated elements of the adaptive immune system as either cells or antibodies released by immunogenic reactions. All of these things, foreign proteins, complex carbohydrates, certain kinds of fats and nucleic acid. Okay, pollens are part of it, even though they're more likely to be haptens, and we have to combine with other proteins and certainly microorganisms. The incomplete ones, too small to be recognized. Short chain peptides, just very short nucleotides, even some hormonal elements. If they attach to a protein, they combine, and then they'll be visualized as foreign. So that's really so you should know the distinction between those two. So what happens, the immune system mounts an attack that is harmful to a person because it attacks self-proteins as well as the hapten. That's one of the problems that we see. And then sometimes we're allergies. So these are interesting. And some of them, there's a little bit of overlap. Okay. A lot of these, like poison ivy, certain kinds of, animal dander is probably more in the typical allergy. Detergents, maybe more in the typical allergy. Cosmetics, again, the poison ivy is more of a resin that's slowly absorbed, and we tend to see a greater amount of cellular response. It's more of a delayed reaction. If any of you, because I am sensitive to poison ivy, I get what's called a fixed drug eruption when I used to get it. So I would always break out here. So it was a fixed location that was probably mediated by cellular immunity. And a lot of people have this. There's things called fixed drug eruptions where people have a specific kind of response to taking a substance. The typical animal dander, detergents and cosmetics, we see more as contact dermatitis. So it has to be on areas that you come in contact with. We saw a lot of them in the foot because you have leather, and when leather combining with moisture or perspiration releases the resins that are used as the tanning process. And, you see it along the straps of sandals, for instance. So I have a vast amount of experience treating that type of stuff. Look what you get. This is why I talk about vast experience. And as I used to say, never start a vast project with half-vast ideas. It's a dad thing. It's a grandpa thing. Don't start. The determinant, the epitope. Does Dr. Maurer use epitope or determinant? Okay. Is there anybody alive out there?
I'm not in the class, so.
You're not. Epitope or determinant?
I mean, I've heard epitope.
Okay. Determinants and epitopes are what basically we respond to. It's that small portion of the antigenic molecule that we form the antibody or the receptor on the cell. So when they have to make a different COVID vaccine, it's because one of those protein spikes on its external capsid has altered and it would no longer adhere to that particular antibody. Same thing with influenza, an alteration of a spike protein. So if it looks like this and all of a sudden it mutates, now it looks like that, it won't bond, it won't attach. That little alteration that it was originally there or modified or mutated is effectively the epitope or the determinant, the part of the antigen that antibodies or the cell receptors bind to. Most naturally occurring antigens have numerous determinants. Okay, different lymphocyte population, different kinds of antibodies. So there's a lot of variation. So you see, that's maybe where we begin to see, if you want to think of certain diseases, why they provoke a wide range of responses, different determinants. They're more complicated. That's why we use simple molecules that don't have immunogenicity. So one of the areas that I had an interest in, and a lot of people do, and we see it particularly, I'm sure the biomedical engineers are like this, is biomaterials. What makes a biomaterial compatible is this. So for years, we used stainless steel. So if we made an orthopedic implant that moved, like a hip or a knee, shoulder, we'd have, because we didn't want to have used a polypropylene or a polyplastic material or a silicone-based material, which was a problem, on one side and metal on the other side, typically coated stainless steel. Because if you had both of the same substance, we tended to get little shards, little fragments that would cause immunological reactions. They used to use, like in breast implants, for breast augmentation mammoplasties, they used to use silicone implants, but the silicone would leak and cause a lot of significant problems that were there. Now, mostly they use units that are put in that they can insert a certain amount of like sterile saline into a non-leaking container. And if there was leakage, it would be unlikely to cause a significant immunological reaction. So biomaterials are all about that. We want to use things that are bio. They use ceramics. They use different types, and the science changes daily. It's such a material science is so fascinating in that regard. So we had a real interest in that years ago. So here's what it looks like. You can have multiple sites, and it's all about the shape. So whether the determinant, in the way they draw it here, is just by shape, whether it's rounded, whether it looks like a spike, whether it looks like the top of my marker, okay, it's going to have a different antibodies it's going to adhere with it. Which brings us to this, MHC proteins. Major histocompatibility complex is what the acronym stands for. They are effectively self-antigens. Variety of these that are covered, that are not antigenic to self, but may be antigenic to others in transfusions or grafts. So that's where the whole idea of grafting, it's another interesting subset of all this that I have a little bit of experience with. One set of important self-proteins are a group of glycoproteins called MHCs, unique to each one of us. Okay? So T lymphocytes recognize only antigens that are presented on certain kinds of MHCs. And you're going to get into MHC1, right? We're going to get into all those that are coming. So the adaptive system involves three crucial types of cells. B cells, the linchpin for the most part, but they do play a role in cellular immunity, of humoral immunity. T cells, cellular immunity, and APCs, antigen presenting cells. Okay, they're a really important auxiliary role. Go back to skin. Remember the dendritic cells that were one of the four cells of the epidermis. You had melanocytes, keratinocytes, you had Merkel cells, which are on the top of the nerve endings. You had this interesting kind of macrophage variant called a dendritic cell because it had almost like multiple tentacles, like a dendrite would in the nervous system, that sampled the environment. What effectively an antigen presenting cell does is it swallows up the invader, or whatever it is for that matter. Let's assume it's an invader for argument's sake. And it grabs it, gobbles it up, and what does it present to the T cell or the B cell? Just the epitope. So that it can make antibodies or cell receptors to it. Love that. It's like, I only had a shirt with a button. I think that's a good visual. None of you I don't see a lot of enthusiasm today. I'm a little perplexed, peeved, bewitched, bothered, and bewildered. Am I?
Off day Wednesday. We'll come back with a vengeance on Friday.
Yeah, oh, you're terrible. It's a great song, by the way. Sinatra does it perfectly. It's from Jimmy Stewart, Kim Novak movie, Jack Lemmon. Ooh, ooh, ooh. Elsa Lanchester. Ooh, Bell Book and Candle. I need to have to look it up. It's a great movie about witches in New York City, 1958. Great song. Oh my God. Maybe it's not from there. It might be from Pal Joey. Okay, I'll look it up. Okay, antigen presenting cells do not respond to specific antigens, but play that role. So we're going to look to natural stopping point here. We have to look at where T cells and B cells come from. Mommy and daddy T and B cells, of course. Nothing. I demand that upon your return from break, that you are more interactive and at least more appreciative of my attempts to educate you.
Okay.
Goodbye. Good Lord, give me a drink.